introduce Dr. David Hong, who, as you can see, is at the Center for Interdisciplinary Brain Sciences at Stanford. Dr. Hong did his undergraduate at Yale, did medical school at Einstein, and then moved west and did his psychiatry and child psychiatry training at Stanford, then at T32 in neuroscience, and now has been 4 years at the center. One of his main focus is on the title of his talk, Sex Differences in Child Psychiatry. So I won't go through mentioning those, but I will mention that the person who invited Dr. Hong here was Fumiko, and then wound up being unable to come and do the introduction. So what she wrote to me was to say he's a truly remarkable clinician and researcher with great insight and creativity, plus simply a fantastic person and team player. Gives fantastic and thoughtful talks. So with that introduction from both me and Fumiko, please welcome Dr. Hong.
Hello everyone, thank you for that kind introduction.
You'll still need to. Yeah.
All right. So yeah, it's too bad I can't see Fumiko. She was a close colleague of mine at Stanford. But happy to come and talk to you guys up here at UCSF. It's not too far. And like Dr. Henry mentioned, I'll be talking about sex differences today. Sex matters. Just to briefly overview what I'll be covering is just really thinking about what is the role of sex differences. You know, I think there's a lot of presumptions that, oh yeah, of course there are sex differences, but it's actually a much more controversial topic than one would think, whether from a social cultural perspective, but even within academic research. And really trying to present why we do need to consider it when we're working either as a clinician or a researcher in this field. A lot of my work in the past several years has been in sex chromosome aneuploidies, aneuploidy meaning an abnormal number of sex chromosomes. And I'll talk about that for the bulk of the work that I'll present today. And then lastly, just really trying to synthesize a little bit of how this research, even though it's still not quite ready for clinical prime time, why it might be relevant to some of the things that we're doing currently in our clinical practice. Is that good?
Better.
Is this better? All right, there we go. So I've been in the sex differences field of research now for some time, and it's, you know, again, like I was saying, there's a lot of these themes that are coming through from science into the lay population, including from your esteemed colleague here, looking at what is the female brain? Is there such a thing as a female brain? And I would be remiss not to mention also the male brain. And this is not something just like people are writing in kind of pop culture literature, right? It's including also theories around Simon Baron-Cohen talks about the male brain, the extreme male brain. So it's really based on a number of philosophies that have been around for decades. But it's not without controversy either. While we might assume there's really what we call sexually dimorphic traits, or true differences between men and women in a number of fashions, other people really interpret this as a bias against males and females in a way that perpetuates, I think, differences across science and in society. And, you know, there's some very succinct and well-written rebuttals to the idea of male-female differences or sex dimorphism in science. And why is this important? Like, why should we actually be discussing it? You know, we could just leave it at that and say there's a debate about it. But in reality, it really permeates a lot of different areas of the work that we're doing and the way that we interact with our patients. On a societal level, it comes up in real-life situations routinely. So we're in Silicon Valley. I'd have to mention our just down the street from Stanford, actually, Google. And I don't know if you guys have been following this memo that's been going around. It's actually quite interesting. This guy got fired recently because he went to a diversity training, and if you go to diversity trainings, it's often about stereotype threat and, you know, the imbalance, particularly in tech companies, between male and female representation in a various number of fields, particularly in coding. And on his way home on the plane, he wrote this kind of 10-page long memo that he posted internally, quoting really what he thinks backs up these differences that they're seeing in the engineering environment. And he posted a lot of really provocative stuff, which included scientific literature to back up his opinion. And some of the things that he listed there was saying that on average, women have certain traits that might impact their role in the workplace. So scientific evidence that they have more openness towards feelings and ideas, a stronger interest in people than in things. And therefore, that might result in preferring jobs that are primarily social or artistic, rather than things that are very concrete, like coding. He picked out different temperamental traits, saying that there's more extroversion than assertiveness, make it more difficult to do things like negotiating for salary or other high conflict situations, and more neuroticism in women than in men. And you might even think, the press asked for a lot of responses from the scientific community, comment on this article, and it's not nearly as homogenous as you would think. There's a number of scientists who said, partly that's right. There are actually some data to suggest there might be male-female differences in some of these things. And then a whole other group of scientists say, this is really outdated, irrelevant data. You have to take it into context, and these male-female differences are not nearly as pronounced as you might think, right? It looks like it's stuck here. Is that moving forward?
Yeah, it's changing the color of your icon.
It's what?
Changing
Oh, there we
the
go.
color of your
Yeah. And so this is an important kind of premise to keep in mind is, really, are these differences valid? And there's going to be a number of things where we say, oh, there's a statistically significant difference between males and females. And probably the reality is, for almost most traits, or for a lot of physical characteristics, a lot of disorders or disease-based things, males and females are probably much more similar than they are different, right? So, in contrast to this whole field of what we call sex differences research, there's been a number of very strong advocates or researchers who are promoting like a gender similarities theory, which is really based on this premise. It's like, who cares if we find these statistical significant difference between men and women, when in reality the distribution across both of the populations is so broad, there's so much variance, that the overlap between these two groups far outstrips anything relevant, right, between men and women. And that's a really valid point, and that's probably true for almost all dimensions. But one thing I will point out is, are there specific instances where it is important to think about sex differences? Are there places where the overlap, either the means are different, or the smaller mean difference is enough to impact some kind of long-term outcome, right? And that's really what we're going to cover today, is to try to unpack a little bit of that, or some of the things where that might be relevant, and to try to think of how we can apply that to child and adolescent psychiatry. And there's probably a lot of places where we really couldn't care, right? Like, even if we leave the social cultural debate aside and just not give it too much credence about how it impacts all these other kind of quality of life measures more broadly, things like height, there are clearly certain traits that are likely sexually dimorphic or show significant sex differences. And this is just like a cool little picture where they basically built a histogram with real people at a small agricultural college in Connecticut in the 90s. And all the women are in white, and then all the men are in the darker colors, the black color shirts there. And you can see, if you look, there's probably a fair distribution across a mean, but those means are pretty significantly different if you break it down by gender. So how do we look at this through the lens of psychology or psychological domains? Melissa Hines, who does a lot of work in this area, wrote a nice summary of some of the findings that captures earlier work in this area. And she found, based on these effect sizes, if you really look at the mean differences and the distribution across a number of psychological domains, there are probably certain areas where the effect size is fairly robust. So core gender identity, this idea like, oh, I'm a boy, I'm a girl. And keep in mind, this is literature that dates back decades, and probably if you look at kids and adolescents now, they're much more gender fluid and might not fit with this really robust effect size. But, at least the earlier findings support that this is a sexually dimorphic trait. Most boys will say, yeah, I'm a boy, and most girls will say, yeah, I'm a girl. Early on, play preference in early childhood is probably sexually dimorphic to a certain degree. And she also found fairly robust effect sizes for things like motor skills or activity level, and then, to a lesser degree, something like aggression. You can't see it as well down here, but she looked also at the literature. Do we have a pointer? A cursor, okay. you don't see it on the screen. But in this bottom right corner, she's also looking at cognitive traits. And cognitive traits are probably one of the most hotly debated topics to talk about something like sex differences. So Janet Hyde and Marshall Lynn, there's some of these researchers who are really promoting what I was talking about as far as really thinking about gender similarities. And what they did is they took a meta-analysis of roughly like 250 studies that have looked at academic performance in boys and girls and looking at math. And this is spanning like 2 decades or so roughly like 1 or 1.5 million students. And they found that there were really no significant effect size or differences in mathematics performance between males and females. They also found a variation, like the level of variance across the male and female population was not that significantly different. So I think we can rest assured, at least for something like the STEM fields, that there's probably good data to support that, at least in outcomes, males and females often perform the same. That's interesting also if you look at it from a cross-cultural or international perspective, right? This is data that's assembled by the OECD. It's like an international consortium of developing nations where they routinely will look at different kinds of outcomes and measures, whether it's poverty or whatnot. And they run this thing called the PISA. It's the Program for International Student Assessment. And what they do is every member nation, there's about 72 of them representing different geographical areas and different levels of economic development, is every 3 years they agree to sample a number of kids in their countries who are 15 years old. So really trying to find the same band across the countries and test how they're going to do in different domains of academic achievement. So they look at math, they look at science, they look at language and verbal skills. And along the bottom here, you can see these are the different countries that participated. And regardless of the performance, roughly, there's going to be countries that are, as a population, have lower performance scores compared to some of the higher developed nations. There is pretty much a broad kind of similarity along the science domain between males and females. So the males are the filled-in circles, and the females are the empty diamonds here. And this is roughly true as well for mathematics performance. Again, males and females grossly kind of approximate each other. There might be a slight increase here in the performance of the male students. So again, at least for these very specific academic achievement outcomes, it doesn't seem like there's a very clear sex difference. One thing that's never talked about, and I go to a lot of these kind of meetings and conferences now, is we don't really often talk about the female performance. So if you look at the reading performance on the PISA, across nations, this is a pretty impressive, I think, difference between female performance, again, the diamonds outstripping male performance on the language measures. So I do neuroscience work. I'm primarily doing neuroimaging and some genetics work recently. And this is the same debate that's been really encompassing some of the things that we look at when we try to identify difference between males and females using MRI. And a number of nice studies have come out in the last 5 years or so where now we're scanning, instead of these small ends of like 10 people, 15 people at a time, we have pretty good cohorts where they're doing like hundreds of kids or thousands of kids. And these are some papers that came out of UPenn and their neurodevelopmental cohort where they tracked a bunch of kids. And they really looked at, are there differences between the males and females in terms of volume or function or connectivity? And they did. They found that there were certain differences in connectivity measures. And again, there's a ton of debate from both the broader community and the scientific community of whether or not these differences really mean anything. And probably the biggest debate about male-female differences when you look at brain size, it leads back to what I was pointing out about height, right? So there's probably a close correlation between a confound of like how tall you are, or how large you are generally, and then how large your brain is. So you have to scale it to the fact that, if all males, like if in general males are larger and have larger brains, the individual structures may also appear larger compared to a group of females if you do this direct comparison. And so that's something that's really hard to segregate between the two. And actually one of the studies, a nice study came out afterwards by Eileen Luders down at UCLA, where she looked precisely at that measure. She looked at female brains of tall women compared to shorter men, so they're fully matched by height. And she showed actually in that study that there's probably very few differences between those two groups as far as male sex differences. So this is something that I think is up for debate and it hasn't been fully answered yet. But a nice study came out this past year looking at that. This is probably up till now the largest single sample study of encompassing both a large population of males and females who got MRI done. And it's about 5,000 individuals, adults between 40 and 70, and roughly split between 2,500 men and 2,500 women. And this hasn't been published yet, it's in bioarchives, but it really illustrates kind of what we're looking at is that there probably are some differences in males and females. So the most universally found one that has been found repeatedly over time is that men tend to have larger brain volumes. So all of the distribution curves, the blue distribution curves. If you look at total brain volume and then just gray matter volume overall and white matter volume overall, it does seem to be that males have larger differences in those regions. But even if you look at the distribution of specific smaller components of the brain, it still seems to bear out that there are sex differences that are preserved. And the general pattern here is we tend to find that males have larger volume and surface area in, for example, the subcortical regions. And you'll see that the men have larger volumes in the amygdala and the caudate, etc. In contrast, women tend to have thicker cortices overall. That was kind of the main finding of this study. And I think this study did a really nice job of controlling for some of these confounds of like the overall sex differences in height, for example. So if you look over on the left-hand panel, they do find these pronounced male-female differences, right? So the blue shading indicates that there's these differences both in volume primarily and surface area, and it's male predominant, that's male. And then on the bottom one, the bottom panel, the mean differences for cortical thickness tends to be with greater cortical thickness in females as opposed to males. And they did correct for the fact that, you have to adjust for total volume. And there are these differences, sex differences, that survive even after that correction. One interesting that they find is that last panel, the variance in differences, it sort of indicates that the degree to which volume is different within any particular group is much more variant in males than it is in females. So it's more broadly distributed, right? As opposed to the female variance ratio here. So why are we even kind of thinking about this? This is all in typically developing populations. Why should we be thinking about this clinically? Like, why is it important for us as all of those you who work with patients? And it's because even if you do it from an epidemiological perspective, we really consistently have been finding sex differences, right? Whether it's in the prevalence of disease, the course of progression of symptoms, and potentially how we approach treatment. There was a big revelation in cardiovascular work when we discovered that maybe women experience heart attacks differently, right? And that had a big impact on how we might approach them clinically. And we may not have yet a similar structure of how we conceptualize or approach this in psychiatry yet. And if you look back historically, a lot of this might be because we just had very biased ways of approaching science and research. If you look at a lot of the earlier studies, it was almost predominantly male subjects. We really didn't study as many women, and we just extrapolate all those findings and assume that it's going to be the same symptomatology, the same phenomenology, and the same treatment models. And what I would really argue, what I hope like everyone will walk out with a message today, is that it is important to consider the sex differences, since it might help us elucidate some of these differences both in the mechanisms that are underlying these disease processes. Are they actually different? And then the sequelae to that, obviously, is might it also impact the way we approach treatment? And if you look, so, I'm a child and adolescent psychiatrist, and in fact, there is a real skew in the way we look at especially in early development, neurodevelopmental disorders are highly male predominant. So, you know, the ADHD, it's maybe 2 to 1, up to 4 to 1. Learning disorders, again, this is a little bit more debated, but somewhere between 1 to 1 and 2 to 1. And then ASD, autism spectrum disorders, really it seems there's a much more pronounced skew where there appears to be a male predominance in effect. And this female protective effect where women seem, or females tend to be less affected. And then if you also consider adolescent psychiatry, this is data from the National Survey looking at prevalence. And it seems like certain disorders, while during the latency period seem fairly equivalent between males and females, there does appear to be emerged now in adolescence a female predominance effect in things like mood disorders, right? Anxiety disorders, eating disorders. And what is underlying this? Should we just kind of take it at face value and be like, oh, like, you know, maybe it's a social cultural difference, right? Like, we assume that girls are more social, so we're less likely to think they might have a problem with social cognition. Or we automatically assume that boys are going to be a lot more of a problem in the classroom, so we let the inattentive girls in the class kind of fly under the radar. It's a complex question, but it probably is all of those things, right? But we'd be remiss not to really think deeply about whether there is a biological etiology also kind of supporting some of this. And a compelling reason to think from that framework is that there's a lot of really robust animal literature to suggest that sex differences are real and biologically mediated. Probably the original kind of study that got this whole line of research in animals underway was done by Phoenix et al. Back in the 50s, where they found really prenatal exposure to testosterone or estrogen had lifetime effects in animals, these very sex-type behaviors. So animals have a just much more programmatic behavioral profile when it comes to mating behaviors, things like mounting, and they have a much more specific way of how they measure that. And when they did these experiments where they're over-rectomized different kind of mice, and then they expose them for a very brief limited period in the prenatal phase to specific sex hormones, even months later, they would find differences in their sex-type behaviors, what they would typically expect to see, and then what was actually happening. And that adds this additional element of thinking like, oh, well, maybe sex differences also have a very time-locked pattern. Like, it's a very complex process where, there are specific parts during development where maybe these sex etiologies affecting sex differences are implemented in a way that is very specific. We happen to have now one of your former faculty who's just really done a lot of great work in this area, Naresh Shah, and he's been also extending this work and really considering, using, again, rodent models, that a lot of the brain is highly populated with sex steroid receptors, right? And we have a pretty limited understanding of how that works, but there's some very specific components that have been implicated in circuitry that we know is related to mood and anxiety and a lot of other psychiatric circuits, including the BNST, for example. So, in this field, the vast majority of research has been around sex hormones specifically, and that's because of a lot of this animal data that came out earlier. And I'm going to make a personal stand of trying to increase more recognition for genetic influences. So it's really gone along these two kind of dimensions. Is like, okay, well, we have these very basic genetics, and probably if there is a biological mechanism that mediates sex differences, it's probably through hormones. It's probably either through the estrogens or it's through testosterone. There's like incredible shaping that it does in these specific time phases. And sadly, I think the genetics have been overlooked, which is really phenomenal and fascinating to me for a number of reasons that I'll cover some of those points as we go along. One researcher who's done some really great work in this area is Art Arnold down at UCLA as well, and he created basically what he calls this 4-core, or I don't know if he created it, but he does a lot of work with this 4-core genotype rat or rodent model, where he separated what gives us male what tracks us into a male trajectory is the SRY gene on the Y. So basically he knocked out the SRY, and so he can now manipulate it to create a genetic mouse that is either it's with a female phenotype that's either XX or XY without the SRY, and then he can also create this other male phenotype mouse that had the genetic sex of either XY or XX with the SRY, right? So he's now separated into these 4 specific ways of looking at what is genetics and what is hormones. And he's done some really nice work in this area showing that there is, in addition to the hormones, and if you go down this SRY, testosterone-driven male differentiation pathway, certain things that we attribute sex differences to. But even in the mice which have a genetic sex that's XX or XY, but not the male phenotype, you'll still see specific traits that are associated with that. And the implication is that genetics is a more critical piece of this than we think as far as how sex differences work. And that might be a domain such as aggression, pain perception, risk for pathology such as neural tube defects or autoimmune function. And even though this is in rodent models, what's really interesting is that if you look at human epidemiology, those are often areas where there are male-female prevalence differences, right? All right, so this is a lot of the work that I've been doing. It really focuses on the sex chromosome, and I'm going to cover a little bit of basic genetics here, just as a refresher, for those of you who haven't looked at this in a while. But they're not equivalent, right? Like, you know, we have 23 sets of chromosomes, and we're diploid, so they roughly, all of our autosomes roughly match, I would say. But the sex chromosomes are in a very privileged category, in the sense that the X chromosome is vastly larger than the Y chromosome. And there's a lot of interesting ideas about how at one point, evolutionarily, they probably were similar. They probably were just like another set of autosomes. And what we call the, you know, it's this idea that like the deterioration of the Y chromosome over millions or thousands of years, and it's resulted into kind of this paltry genetic chromosome here, right? So the X chromosome is more than 150 million base pairs. It's probably 5% of your total genome, and then the Y chromosome is only about 50 and only 2% of genome, and has 1/10 the number of what we think the coding genes are on the X chromosome. So you would think this gives an immense amount of privilege to women in the sense that they have 2 X chromosomes with this really robust genetic material, and guys only have 1 X and then 1 tiny little Y. And we'll talk about their, you know, nature has taken care of that for us a little bit with dosage compensation. But you know, this is also an interesting way to approach potentially like this big difference in genetic material. Can that underlie maybe some of these differences we're seeing in between men and women and these sex differences? So this work I've done with Alan Reese during my postdoc. He's been in sex chromosome field for a number of decades now. And the 2 disease models that we were looking at for the last several years is Turner syndrome, which is the only monosomy that's compatible with life in humans. And that's where 1 X chromosome is missing. And so, you know, it's actually much more common than we think probably, but it's not fully penetrant, so it's not clearly, I think this data is based on older Scandinavian data. So this is a condition where you only have 1 rather than 2 X chromosome for Turner syndrome, females with Turner syndrome. The other syndrome we look at is something called Klinefelter syndrome, and there's 47 XXY, where males, they still have an additional X chromosome. There's a non-destruction event, and they carry it in most of their cells. And this is an extremely common disorder, and it's very highly variable, the phenotype. Like a lot of people, it's not penetrant at all, and they may go through their entire life without knowing that they have Klinefelter syndrome, and it may not affect them in a meaningful way. But it is one of the most common, again, causes of gonadal dysgenesis and you know, low testosterone. So it's one of the first things they check in fertility exams if you're having trouble. Like that's one of the screens that they'll do. So we were looking at these 2 groups, and it gives us now this model of thinking about sex chromosome dose, this idea like, oh, you know, typically developing women have 2 X chromosomes and typically developing men have 1. And then what's going to happen if you extend that model further on either side using aneuploidy, right? So now you have the Turner syndrome cohort where they have 1 X chromosome, and you have a Klinefelter group that has 3 sex chromosomes, 2 of which are the X chromosome. And if you look at the neurocognitive phenotype, they're really both roughly in the normal intelligence range. So, if we consider 100 to be the population mean, they're probably like 5-10 points below that in full-scale IQ, right? So it's still what we'd consider in the general range of normal. And what's driving probably that little dip is that it's an uneven profile. And typically, like the classic Turner neurocognitive phenotype is that they have a preserved or enhanced verbal IQ, and what's driving this couple points going down is that they have a lower performance IQ or nonverbal IQ relative to their peers. And then, in contrast, the Klinefelter population, they have the opposite pattern, where they tend to have a relatively preserved performance IQ and then a lower verbal IQ, or they both tend to have specific kind of social cognitive deficits, although they may differ in some degree, and they tend to have differences in executive dysfunction compared to control cohorts. Similar to what you'd see in these IQ domains, girls with Turner syndrome often have things like visual spatial impairment or arithmetic difficulties, whereas boys with Klinefelter syndrome might have verbal processing impairments. And so again, you could see if the mean is 0, looking across, I also included XYY here, just reviewing the literature, you can see that there is a fairly typical pattern of whether you have a triploid sex chromosome genotype, so 3 versus 1, where there seems to be some level of inverse relationship between them that would match with chromosome number. And the clinical phenotype in these groups really match up with some of the areas that are associated with these neurocognitive domains, right? So both of them have executive function, and ADHD is highly disproportionate in these populations. So it's 18 times increased in ADHD, and then or in Turner syndrome, and then 30 to 50% of boys with Klinefelter syndrome meet the criteria for ADHD. Dyscalculia, or mathematics learning disorder, is highly represented in the Turner syndrome population, 50 to 79%, whereas language-based disorders or dyslexia in the male population of 50 to 75% again. There's also a higher risk for autism and schizophrenia in KS, and then in Turner syndrome, it's more social anxiety and social communication issue. So a lot of the work that we've been doing in the past years during my postdoc is we looked at the sample of prepubertal children. So really before they started hormone replacement therapy. It's not uncommon for Turner girls to be diagnosed very early on because they have very characteristic physical sequelae that's associated with the condition, which tends to be short stature, wet neck, and things like that. So girls with Turner syndrome really get picked up very early in life, and so they'll be tracked in the medical system for some time. In contrast, a lot of boys with Klinefelter, they fly under the radar, and so they're sometimes just clinically referred when there's some problem going on. But we very specifically targeted both of these groups because neither of them will get either estrogen or testosterone until around the time that they're supposed to hit puberty. So we focused on this group of kids who are between 6 and 11 roughly. And this is actually a study I did with Famiko. We compared the two groups as far as structural MRI patterns, and it was remarkable to find the differences between the two groups were really complementary in opposite directions. So on the left-hand side, you see this is the area in the parietal occipital regions or the somatosensory cortices where you see the Turner syndrome have lower cortical volume compared to typically developing females. So they have one X chromosome and they have lower volume in this region. In contrast, in almost the same exact region, the Klinefelter boys who have 3 sex chromosomes have larger volumes compared to the typically developing XY males. And then if you look in another contrast, in the temporoinsular regions, in the Turner syndrome group, they have larger volumes in that area compared to typically developing females. And then, and again, almost identical kind of pattern in these two separate cohort comparisons. The boys with Klinefelter syndrome have reduced volumes in those areas. So, we're finding converging, just by sort of chance, we're looking at these things separately, but we're finding these converging data suggesting like the expected pattern does seem to follow a linear kind of dose response curve, right? So in the same regions, you might have some areas where there's one, you have one less sex chromosome or one more, and they have the flip pattern, which is what we would anticipate. And that's just another illustration of that. And so we're really trying to dig a little deeper and say, oh, well, okay, this is kind of unexpected, and in the sense that we weren't looking for this specifically, but it is what we found. Like, very few people are studying both of these cohorts simultaneously. And what is really underlying that? Like, what can we look at as the reason that this is happening? And speaking back to kind of the debate about what's hormones and what's genetics, really in Klinefelter syndrome, you rarely have testosterone deficiency in early adolescence or childhood. The involution of the seminiferous tubules that cause testosterone deficiency typically doesn't occur until the onset of puberty. So for most boys with KS, they will have relatively normal pre-pubertal levels of testosterone. And so that's the population we're studying, right? So our assumption, just by that, like the cohort that we gathered, is that this isn't an effect that's driven by hormones alone. Like, we're finding these really robust neuroanatomical differences, and we can't attribute it just to hormones. We didn't measure hormones as part of our study, but the presumption in the broader literature is that this is probably driven by a genetic effect, right? And so, you know, it led us really to think about, okay, what's happening on the genetic level here? What's happening with this sex chromosome dosage effect? And again, just kind of to illustrate that, it's either just by sex chromosome number, you have 1, 2, or 3 chromosomes, with typical development having 2 of each chromosome, and then 1 and 3 on the other side. And then again, this hormone status, which we think is maybe not explaining all the things that we're finding, at least in the, specifically in the Klinefelter population, but still having these differences. And I talked before about the fact that, you know, males and females have very imbalanced genetic material, and evolution has taken care of that for us in the sense that they wanted to make sure we're fairly equivalent, just so as not to create any disadvantage, I think, right? And the way we do that is we have this automatic kind of inactivation effect. So in typical development for women, one of the X chromosomes always gets turned off. And so all women are chimeric or mosaic in the fact that there's this random inactivation pattern. And, you know, there's emerging research to suggest that maybe it's not as random or as equivalent as we think. But, you know, roughly about half the cells in women, typically developing women, will have the paternal X turned off, and the other half will have the maternal X turned off. And then it puts us on a level playing field, right? So now both men and women have roughly 1 functioning sex chromosome. And it also should, in theory then, balance against something like Turner syndrome, where there's 1 functioning X chromosome, which obviously doesn't get turned off. And this was nicely illustrated for a physical trait of height. I mentioned before that Turner syndrome, it's characterized by short stature, and they did this actually back in the 90s, and, you know, where they identified a specific gene on the X chromosome called SHOX on the distal tip of the short arm, which is related, you know, stature is obviously multigenic, but it identified that SHOX is one of these genes that are on the X chromosome that contribute to height. And it matches up again. The inference is that there's this dose-specific effect. So girls with Turner syndrome who have 1 X chromosome tend to be shorter than their female peers. And then boys with Klinefelter who have 3 copies now of the SHOX tend to have taller height. So, you know, again, the dosage that you have influences the activity of that gene. And it's because SHOX lives in this particular region, the ends of the sex chromosomes, called the pseudo-autosomal regions that don't get turned off, all right? Because we would think like, oh, well, okay, like Turner syndrome, if you have 1, then why do they have all these physical differences? And probably the genetic premise behind that is because actually there are certain parts of the sex chromosome that don't get inactivated, so that might be driving a lot of these effects, right? Then a lot of the interest will be then in these distal regions, or these pseudo-autosomal regions on the sex chromosomes, which live on the tips. And there's also a whole bunch of other chromosomes on the X in women that don't get turned off either. They're called de-escapee genes. So you can see on the sex chromosome genes, like a lot of them are implicated in cognitive function, right? Like there's a whole bunch of regions in the PAR1 and the PAR2, but also distributed across the X chromosome that have been found to be affected in a number of disorders that have a cognitive phenotype. So I think there was a lot of interest then in saying like, oh, well, you know, can we find this and replicate this in Turner syndrome, right? So can we map, for example, some of these things that we're finding in the Turner syndrome population onto what we're seeing from a genetic perspective? And there's this early deletion study that really looked at not just people who had all the cells were deleted, but just certain portions of the X chromosome were deleted. And as they slowly track which parts were deleted, which one of them showed this classic phenotype that we tend to see in Turner syndrome. And it appeared that maybe there's a correlation to the distal portion of the X chromosome in the pseudoautosomal region. And that was again repeated with other kind of domains in Turner syndrome. And you know, again, but it's a tricky question because it wasn't replicated in another study. Someone looked at it in Klinefelter syndrome and also found that same region. Interestingly, the part that's deficient in Turner syndrome, like there's less production in that area, causes a particular phenotype. It's overexpressed in Klinefelter syndrome, causing the opposite phenotype. So a very interesting kind of idea that maybe there's specific genes in the sex chromosomes that's driving a lot of these effects. So, you know, the next part I'm going to talk about the genetics, and you know, the hope was like, oh, well, we're just going to have this really parsimonious kind of theory that ties all this together, is that there's going to be 1 or 2 genes on the X chromosome that explains everything that's happening. And I think that's really what everyone would hope for, but obviously it's never that simple, right? And it's not as if we're the first ones to think about this. We're doing it in sex chromosome disorders, but you know, people have done it in other kinds of areas, like including, for example, autism spectrum disorders, and seeing how much the X chromosome is implicated in that. And when they really looked at it, you know, they didn't find a single SNP that could explain the autism phenotype based on SRS scores that were identified on the X chromosome. So in reality, it's probably not a very simple story. But basically what I did with some of my collaborators, Alex Urban and his postdoc Zheng Long down at Stanford, is we took a sample of all these kids and we did RNA-seq. So what that does, it measures roughly the activity of these genes that live on the X chromosome. And this is just a pictograph showing like the level of expression generally that differs between girls with Turner syndrome, XO, versus typically developing girls, XX. So the amount that it varies from the red line is roughly the differential expression, right? And you know, a nice proof of concept and a sanity check for us was that EXIST, which is the X inactivation gene, it's highly, highly much more expressed in typically developing girls. So that makes complete sense. They have 2 X chromosomes, they have to turn one off, and so the gene that's responsible for that is highly active, and it's essentially not active in the Turner syndrome group. So that's very, very reassuring. So that's near the centromere there. You can see the EXIST gene. So that's highly differentially expressed. And it seems like maybe there's a number of other genes on the X chromosome that also might be differentially expressed. And they tend to cluster in PAR1, right? So they're or just near the PAR1 region. So again, that was very reassuring. And we found the same profile if you look at the differential expression from the X chromosome in boys with Klinefelter XXY compared to typically developing boys. Again, typically developing boys with XY have no reason to inactivate a gene, but Klinefelter boys do inactivate that second X chromosome. So again, nice to see this highly differentially expressed EXIST gene activity. And again, it seems in PAR1 there seems to be a fairly high number of differentially expressed genes. And we tried to look at these PAR1 genes to see if they're implicated, and we weren't, you know, we haven't correlated fully to the phenotype, but we weren't fully seeing that there's a direct correlation between these gene expressions and some of the things that we find. And so we stepped back a little bit and said, okay, well, you know, maybe we're not going to find a single gene on the distal end of the X chromosome that's going to explain the entirety of like the Turner neurocognitive phenotype or the entirety of the Klinefelter phenotype. And this is basically a Manhattan plot where you look at it not just in the X chromosome, the sex chromosomes, which is 23, but across all of the chromosomes, right, in the genome. And you can see there's a whole number broadly across the entire genome where there's differences in expression. And it's not just limited to the sex chromosome. And the same is the case for XXY males compared to XY males. There's across the transcriptome a whole number of genes that are differentially expressed. And this is, you know, if you look at a lot of studies out there, sadly, they just simply exclude the X chromosome from analyses because they don't know how to deal with it. Like, it's a little bit inconvenient. So there is like a vast majority of these studies simply won't include differential expression from the sex chromosomes as part of their data. And so we're really lacking a very clear basis for understanding what's happening in this area. And so we kind of sat down and thought like, oh, well, what can we do with the idea? Is there anything that's tying these different themes together? We've shown in neurocognitive phenotype and we've shown in neuroanatomical phenotype that there might be a specific dose-related response, right? So the premise underlying that is depending on the number of sex chromosomes or X chromosomes, and then more specifically genes within the sex or X chromosome, we have this response that is dose dependent. And we have basically these two group comparisons where we just group them all together on the basis of having 1 versus 2 X chromosomes. So in Turner syndrome, it's 1 X chromosome versus XX. In XY versus XXY. And then also in typically developing males and females, it's XY versus XX. So in each of these 3 comparisons, we looked at all the differentially expressed genes based on having 1 versus 2. And then we looked at all those differentially expressed genes in this Venn diagram and say, which of these are shared? How many of these differentially expressed genes between these pairs overlap across all 3 groups? And that left us with these 31 in the middle, right? And it almost exactly follows kind of the anticipated pattern of this idea that there's a dose-dependent effect based on the number of X chromosomes that you have, right? So in the comparison where it's XY or TS compared to XX, then you know, both these across all, this is across the entire genome, not just the X chromosome, it's roughly halfway the anticipated differential expression. And then in XXY, where they have an additional X chromosome, the differentially expressed genes show a pattern where they're overexpressed, again, above what would be anticipated for the typical level of 1. And so, you know, we were thrilled with this result because it was really interesting. I mean, you know, as far as we're aware, this hasn't really been published, in the idea that there's all these effects, not just in the sex chromosome that have a dose-dependent effect, but it's something that broadly covers all 23 chromosomes. So not all 23, not all the autosomes and sex chromosomes are represented in these 31 genes, but the implication is that there might be a genetic effect that downstream affects broadly the level of expression across a number of autosomes, something that, you know, simply I don't think is part of a big part of the conversation right now and how we approach things. And then that provides sort of a genetic basis for maybe thinking of why we have sex differences across a number of regions and genetic domains that aren't necessarily on the sex chromosomes, right? So for my K data, like, we also recruited this population of girls with Turner syndrome who have a mosaic karyotype. So I've been talking mostly about kids who are monosomic, in the sense that we assume that almost every single cell is either Klinefelter or Turner karyotype, meaning XO or XXY. But the reality is, if you have this non-disjunction event later in development, that, you know, it's going to be a chimeric. So a certain number of cells will have the typical genotype of XX or XY, and then the other proportion will have, you know, either the Turner or Klinefelter one. And so we did this in Turner syndrome, where I recruited, you know, 20 or 30 girls who had a mosaic karyotype. So they all have differing percentages of a classic Turner phenotype. So using FISH, we measured some of the girls had like 25% cells that are XO and 75% XX. Some of them have 50-50, equally distributed, and some have 75%. And if you go from a dose dependence model, the idea is that even in mosaicism, if you regress them all to, you know, a distribution, you might anticipate the more normal or XX cells that you have represented, the more normal also the phenotype might look. And in fact, we found that for a number of the neurocognitive domains that are implicated in the differences between Turner and Klinefelter syndrome. So we put a bunch of kids who have monosomic, so those are all the zeros, the dots, and then typically developing girls who are 46XX, those are the dots with 100. And this is all the data, but you can see there are certain areas that tend to center around visual spatial skills. So this is NEPSI domain scores, things like picture concepts, or somatosensory skills, things like imitating hand positions, visual motor precision. these are all show that depending on the level of mosaicism, the percentage mosaicism between 0 and 100%, they're going to follow this pattern that seems to be dose dependent. So it's really nice for us to find some kind of confirmatory kind of premise that even mosaicism might modify some of the things that we're finding that would fit with our genetic premise. All right, so I just covered a lot of stuff about sex chromosome disorders, and you're probably almost never going to see any patients or kids with sex chromosome disorders. So how is this relevant to the larger field? Like, what's a way we can really think about this? And what I hoped I convinced you of a little bit is just really think about how sex chromosome aneuploidy research gives us some insight into just sex differences more largely, right? To me, it's important that a lot of the neurocognitive profile domains that we see in sex chromosome disorders happen to overlap with childhood disorders that show some of the greatest sex differences, right? Whether it's ADHD or autism. Those are the same domains that tend to be affected in sex chromosome disorders. So maybe it's a great disease model for us to think a little bit more about how does sex play a role as a biological variable in those conditions? And to really show that maybe there's an alternative to this idea that a single gene is disordered in sex chromosomes, but in fact the idea that we have these complex genotypes that affect the entire transcriptome broadly. And in reality, the work that still needs to be done is really seeing how all of these things interact with epigenetic and gene-gene and gene-environment interaction effects to better understand what's happening. One of the things when we think about sex differences in psychopathology is that probably, when I see most of my patients, I don't make a strong discrimination between who's male or female in my practice. Like I'll still follow the same algorithms in how I diagnose and how I treat them. We don't have anything in the DSM that says specifically consider this as a diagnostic criteria in boys versus girls. We don't have any treatment algorithms that say these girls should be on X medication and boys should be on Y medication. We'll universally apply a lot of the same principles to our populations, rightly or wrongly. And what I want to do is challenge why we're not thinking more about that, especially in these disorders that have huge kind of variances in male-female sex ratios for prevalence, right? And there's some researchers who work in these areas where they really study the difference between boys and girls and how it looks in terms of phenomenology. Steve Henshaw over at Berkeley has been following the longitudinally these cohort of girls with ADHD for many years. And, he says one of the things that he's finding is that there might be really specific differences, right? Like in the sense that girls with ADHD have all the problems associated with ADHD. Plus they might have more problems around self-harm and emotional discontrol. And so, it's almost as if they're less likely to have ADHD, but when they have it worse. They have it plus, right? And Tom Frazier and even Kevin Pelfrey, who does a lot of autism research, is finding something similar in autism, where he says, look, autism is way more prevalent in males. Way fewer females are likely to be identified. But the females that are identified, they tend to have fairly severe forms of autism. Like it seems like the characteristics of it is that you have this female protective effect, but once that threshold is crossed, you're going to have it worse maybe than your male peers. And so, again, what is underlying that is we need to figure it out in a more specific way. This is work, again, on our Turner syndrome population that's been expanded following them through puberty. But Tamar Green, who is a phenomenal junior faculty in our center, who will be coming to you guys, I think, in a couple months, she's very interested in ADHD and has also applied some of the work that she's been doing in Turner syndrome. And she looked at the BASC scores, so looking at attention and hyperactivity. And if you're looking at the difference between green, neurotypically developing kids, the red, which is the girls with Turner syndrome, and blue, which is idiopathic ADHD, she found that, again, not surprisingly, there's a much higher number of girls with Turner syndrome who are affected by ADHD. So this really replicates what was found earlier. But she went a step beyond that, and she emailed the BASC psychological, she asked for the actual normative data from the BAS company and said, hey, can you give me all your idiopathic and normative ADHD data? And they sent her to the data so she could do this analysis. And she compared now, she broke down the Turner group into the girls who have ADHD and girls with Turner syndrome who don't have ADHD. And in fact, not surprisingly, just because you have Turner syndrome, it's not equivalent to having ADHD. The girls with Turner syndrome who don't have ADHD are like typically developing kids without ADHD. And then she looked really at then now, the girls with Turner syndrome who have ADHD compared to idiopathic boys with ADHD and idiopathic girls with ADHD from these normative BAS samples. And in fact, the profiles associated with the Turner syndrome is much more similar. There was no differences between idiopathic boys with ADHD, and there were differences with idiopathic girls with idiopathic ADHD. So it seemed like, with Turner syndrome, yes, they have ADHD, but if you dig a little bit deeper in an exploratory way, it follows a male profile of ADHD, suggesting that, again, there might be sort of this male-specific type to it related to, potentially, again, the sex chromosomes. So I didn't touch on hormonal effects at all, and I'm wrapping up on time here, but this is, again, something that we're very interested in, that I think we're really doing a disservice to our field by not considering it. We have almost no hormonal research in our area. They're incredibly important signaling mechanisms that we don't talk about. This isn't the case in a lot of other fields, like endocrinologists routinely work with hormones, and it's curious why it will often be like, oh yeah, sex hormones or sex steroids and leptin, and we know people get manic when they're on corticosteroids. We acknowledge all the time that these have really strong links to psychiatric phenomena, and yet we don't have a very comprehensive program. We're trying to understand really well neurohormonal effects in child psychiatry. So as a last sort of plug, I am collaborating with Steve Rosenthal, who's one of your ped endocrinologists here at the Gender Center, and my colleague Tandy. We're looking more at some of these things in adolescence and early pubertal transgender youth. So really trying to understand some of these processes as kids undergo cross-sex hormone therapy. So it's not quite underway yet, but I may ping you in the future to think about if you have these kids you're seeing in your practice to refer them to us. So just to thank everyone in our lab at the Center for Interdisciplinary Brain Sciences Research. A lot of the work, again, has been done by Lindsay and Antonis and Sharon, the RAs, and then Tamar, who's a young junior faculty in our center, who's coming to you guys. And then I have to thank all of my collaborators who I do my genetics and endocrine work with. So thank you very much.